Good morning for me, and good evening and good afternoon, or everybody. First of all, I'd like to thank organizers, Chris and Giovanni and Diana, for giving us a chance to talk to you about one of our projects that brought us to the field of sphingolipid and ceramide. And I've been coming to this Sphingolipid bi-weekly Zoom meetings over the last couple of months, and talks are just fantastic. And I'm really looking forward to meeting each of you in person sometime very soon. So today, I'd like to talk to you about the unfolded protein response pathway, or UPR, which swings into action to generate or adjust the overall functional capacity of the endoplasmic reticulum. Now, ER is the one of the biggest compartments in the cytoplasm of the eukaryotic cells, and as a gateway of the secretory pathway, ER plays very important roles. Almost all the proteins that are secreted outside of the cell, for example, growth factors, hormones, and cell surface receptor kinases, channel, all these proteins, as they synthesized, they have to actually enter into the lumen of the ER as linear unmodified polypeptides. And inside of the ER, those polypeptides will then undergo a variety of maturation steps, including chaperone-assisted folding, modifications, glycosylation, disulfide bond formation. In some cases, subunits have to come together in order to make the mature, fully folded form of the protein before they exit from the ER to headed to the cell surface or outside of the cell. Turns out that those proteins we collectively called secretory pathway proteins, but consist about 1/3 of the total proteome of eukaryotic cells. So this is not a small job for the ER. And speaking of not a small job, the synthesis of almost all cellular lipids initiates here on the surface of the ER. So that's also a big job for this compartment. Furthermore, ER plays a variety of different functions, some of which are shown on my slide. But this is basically a very much multitasking, busy compartment. But turns out that those activities are not static during the lifetime of the cell. In response to a variety of different conditions or environmental developmental cues, the activity has to change. So this slide is one of the good examples where mature B lymphocyte is becoming antibody secreting plasma cells in response to, in exposure to antigen. And this actually whole area of the membrane part is all ER, and in anticipation of secreting huge amount of antibodies, somehow cell knows to expand the ER in order to keep up with the demand of secreting massive amount of secreted antibodies or immunoglobulin chains. And now we know that a major part of this is mediated by the unfolded protein response pathway. So again, in response to ER stress, throughout my talk, I'm going to use this term ER stress, collectively referring to increased demand of ER functions or the accumulation of unfolded proteins. The UPR initiating components, IRE1, PARC, ATF6, in mammalian cells activated. I will talk about these components in a few minutes, but those activation leads to the induction of downstream signaling cascades. And that allows ultimately transcription induction of genes coding for ER chaperone, protein folding modification, folding or modification enzymes, and lipid biosynthetic enzyme or metabolic genes. And together, this will allow the re-establishment of the ER homeostasis. Now, UPI is conserved all the way across eukaryotes, so starting from single-cell eukaryote budding yeast to the mammalian cells, where at least 3 UPI initiating components are present on the membrane of the ER. Now, what I'm going to talk about today is this ATF6. ATF6 is a cryptic transcription factor. What I mean by that, it has a single-span transmembrane domain, and with the luminal portion of the protein, ATF senses or measures the levels of unfolded proteins, and that leads to activation. And activated ATF6 then moves from the ER to the Golgi, where 2 proteases are waiting to cleave this protein within the transmembrane domain and releasing part of the protein into the cytoplasm as a soluble protein. And this portion contains transcription activation and nuclear localization signal, so it goes into the nucleus and activates UPR target genes. Now, what I'm going to tell you today is that ATF6 can be activated by sphingolipids, very specific ones, by a mechanism that's unique to the previously known activation of ATF6, so not through this so-called stress sensor domain. And one of the surprises was that this activation leads to the transcription induction, but the target genes turn out to be a little different from ATF6 when it's activated through the stress sensor domain. So how did we stumble on those actually findings? In order to tell you that, I first need to take a little detour and tell you about cell cycle. So we know that the genome is important, and replication and sister chromatid separation will have to take place in timely manner in order to generate 2 cells with a complete set of genome. Now, several years ago, working in budding yeast model experimental system, we found cell cycle checkpoint for dividing ER. And it turns out that this pathway or this checkpoint is one of the very first checkpoint for regulating the division of the cytoplasmic components, including organelles. So we're very excited about this, and we call this cell cycle checkpoint as ERSU, ER stress surveillance checkpoint. Now, what it is that when ER is too small or not so functional to divide into 2 cells, essentially inheritance, giving ER into the daughter cell stops. And ultimately, this results in block of cytokinesis. So cell cycle stops. And in the meantime, UPI is being turned on, and this will allow all the functional capacity or even size of the ER to adjust enough to undergo divide into 2 cells. And when that occurs, cell cycle block is released, and cells can go back into the cell cycle. So this is actually classical cell cycle checkpoint that we hear a lot about for DNA. But even more exciting to us was that recently we found phytosphingosine is induced by ER stress, and this phytosphingosine is the activating signal for the ERSU cell cycle checkpoint. Now, I don't have time to actually go over this in detail today, so I hope I'll have a chance to talk to you about that sometime near future. But this actually finding raised question that if ER stress in mammalian cells also increase any of the sphingolipid or ceramide species. And to ask this, we used the pharmacological agents that are very well known to induce ER stress by different mechanisms. So for example, sapsigagen releases calcium from the ER and thereby inducing UPR. DTT disrupts disulfide bonds, so accumulates unfolded proteins, again in the ER. And treating human embryonic kidney cells, we found that during the time course, we saw the elevation of dihydrosphingosine, DHS, during this time course. We were surprised to find that all the downstream, more actually the abundant lipids, ceramide or sphingomyelin, sphinganine, they are actually not, their levels are not much changed during this treatment. Similarly, when we induce ER stress by different mechanism, by DTT, again, we saw increased level of dihydroceramide. So with this audience, I don't think I need to show this, but both the DHS and DHC are produced on the surface of the ER and are products of the biosynthetic pathway. So we thought that this increase has any physiological relevance or functional importance. And to ask that, obvious choice would be to look at the mammalian ARSU pathway, cell cycle checkpoint that I just mentioned that to you. But we are actually just starting to understand the mammalian cell cycle checkpoint, ARSU pathway. So while we are doing that, we thought mammalian cells actually have two extra UPR components that are not present in budding yeast cells. So we thought that maybe we should check actually UPR pathway is altered by those sphingolipids. So to do that, we used ATF6 reporter where GFP is fused to the cytoplasmic portion of the protein. And with this, initially the ER localized ATF6 moves to the Golgi and then ultimately enters into the nucleus. So by monitoring whereabouts of the GFP, we can assess activation status. Furthermore, by quantitating this fluorescent level, particularly the nuclear localized GFP, we can again assess the kinetics of the activation and also the extent of overall level of ATF6 activation. So using this, we found that when we exogenously add DHS or DHC to mimic the increase that caused by ER stress, we found that this reporter accumulated in the nucleus at the extent and the kinetics very similar to either DTT or the sapsigagen. So this actually was exciting to us that, well, maybe actually this is doing something. So we thought maybe other UPR components, the PARC and ILE1 might be also activated. So for PARC, we looked at the phosphorylation of EIF to alpha. Actually, I should go back. This phosphorylation is carried out by PARC kinase. And phosphorylated form of EIF to alpha diminishes translation initiation. So essentially, this will actually repress the global translation. And that makes sense. If you have a lot of proteins in the ER to fold, you may not want to keep making a lot of proteins. And by monitoring phosphorylated form of EIF to alpha, by the following either sapsigagen or DTT treatment, we can detect robust level of phosphorylated EIF to alpha, but not with DHC, and not shown here, but with the DHS as well. Similarly, when we looked at ILE1, this component, by splicing of XBP1 mRNA, by RT-PCR across this intron, IRE1, activated IRE1 actually splices this message. And with the subsigagen or DTT, again, we can see the robust change of the smaller spliced form of XBP1. But with the DHC, and not shown here, but DHS, essentially, we didn't see the appearance of smaller spliced form of XBP1, suggesting that IRE1 is not activated by DHS or DHC. So this suggests that those sphingolipids are specific to ATF6. So question is then, how do they activate ATF6 then? Initially, we thought that maybe those sphingolipids cause somehow accumulation of unfolded proteins in the lumen of the ER. So using folding reporter, which is a fusion protein between the vesicular stomatitis viral glycoprotein fused to GFP. This VSVG is a cell surface protein. So in order to fold and get to the cell surface, it has to transit through the for folding and modifications. So by also blocking this protein from actually getting out from the ER after folding, so we have all the fusion protein accumulated in the ER, we can assess whether this fusion protein is folded or not folded, because we have antibody that against the properly folded form of VSVG. So if you do immunofluorescence with this antibody to look at VSVG portion of the protein, you can see the signal, pretty good, actually robust signal. But when we treat cells with DTT, now the immunofluorescence actually goes away, signal goes away, but fusion protein is present in the cell when we look at the GFP portion. Now GFP portion is small and relatively well actually folded. So even in the presence of DTT, it can still emit the fluorescence. When we add DHS or DHC, we found that this antibody that recognizes the folded form of VSVG still recognize this fusion protein, meaning that this is actually mostly properly folded in the ER. So this suggests that folding capacity in the ER may not be changed too much, but because this is one protein and also viral protein, we wanted to actually make sure, and we performed a variety of different experiments. Now, due to the time, I don't have actually time to go over every single data, but I'm going to show you two actually key experiments. One of them is the association of this major chaperone for ATF6. Now, we know that it's been known that the luminal portion of UPR initiators, both ATF6, IRE1 PARC, they're all bound by this chaperone. But when you induce ER stress and increase levels of unfolded proteins, chaperone leaves from there and attend to these unfolded proteins in order to fold them. So if we look at the BIP association with ATF6 by co-IP, initially we can see pretty robust association. Co-IP BIP comes down with the ATF6. But when we treat cells with subsigagen to induce ER stress, the BIP dissociates, and this will be, BIP is actually working on, presumably working on unfolded proteins. With DHC or DHS, pretty much we saw the BIP still remain associated with the ATF6, suggesting that not too much unfolded proteins are present in the cell when we add DHS or DHC in the cell. Now, by the way, we actually confirmed that addition of those lipids into the cell media cells will take up, and they actually lead to the get to the ER. And we actually deleted luminal portion of IRE1 that's been mapped to be important for recognizing unfolded proteins. So deletion like that obviously will inactivate ATF6 from recognizing unfolded proteins. So that's what you're seeing in the presence of the deletion. It's no longer producing activated form of ATF6. However, this deletion did not have any impact on the activation by DHC, and not shown here, but also DHS. So together, those experiments started to suggest that maybe not the ER luminal, so-called ER stress sensor domain is important, but perhaps transmembrane domain of ATF6 is somehow recognizing DHS or DHC levels. Now, when we were looking for any kind of sphingolipid binding motifs, we found this actually very beautiful work that on the P24 COP1 machinery protein, and within its transmembrane domain, the authors found that there's actually 9 amino acid motif that's actually specifically binding to the sphingomyelin. And also they performed variety of mutagenesis and know that some of those residues are critical. So based on that, we looked at the ATF6 transmembrane domain, which is highlighted in yellow. We found actually not exactly like P24 sphingomyelin binding site, but somewhat similar, particularly those actually residues conserved or thought to be important for binding to sphingomyelin is also present, and their spacing between them is kept, maintained. And furthermore, when we looked at ATF6 from other species, we found that these residues are actually conserved. So we thought, well, maybe we can see what happens. And so we decided to change what individual amino acid one at a time, and those single amino acid changes of ATF6 was tested for whether they can actually produce nuclear form of ATF6 when we add DHS. And what we found is that those leucine or isoleucine, 5th position, and the last one, tyrosine, when we change that amino acid to either phenylalanine or cysteine, the level of the activation of ATF6 went down, whereas other single amino acid changes did not seem to affect greatly to that. This is also true with the DHC. But we didn't actually destroy the protein. We were very worried that by changing amino acid within the bilayer, maybe we're just creating unfolded proteins. And when we treated with DTT or thapsigargan, those actually single amino acid changes, they are able to activate ATF6 about quite actually robustly. So it appears that this transmembrane domain motif is sensitive to the DHS and DHC. Does it bind to those lipids? Well, we used radiolabeled DHS and performed the binding assay with the wild type ATF6. We can see the binding in concentration-dependent manner, but not with the mutant that actually did not generate activated ATF6. And binding is specific because we can compete this binding with the cold excess of DHS, but not by unrelated lipids like cholesterol. So we were very excited about this. And perhaps that ATF6 transmembrane domain has a unique binding site for those early product of biosynthetic pathway of the sphingolipids. But before we go on and excited about it, we wanted to make sure that nuclear actually generated the nuclear form of ATF6 is active as a transcription factor. So we performed genome-wide transcriptome analysis. And when we looked at ATF6 target genes that are previously defined, and particularly those genes are related to some sort of lipid function, either on the pathway, we found that their transcription is robustly activated. Surprise came when we looked at the genes for chaperone or protein folding components. Their transcription doesn't seem to be robustly activated by DHS or DHC. Again, I'm only showing you just DHC, but this is also true with DHS. So it appears that there are two independent ways of receiving signal. One is through transmembrane domain. And with these mutants, you can still activate by luminal stress, but luminal mutation will abolish the ability to activate this protein. In contrast, those mutations within a binding motif will actually diminish ability of ATF6 to be activated. But this luminal domain mutant, because it has a wild type transmembrane domain, it can still respond to DHS or DHC. So perhaps that based on the transcription output, when you activate through this transmembrane domain by sphingolipids, these are more catered towards taking care of lipid synthetic needs or the lipid-related functions. Now, when we were thinking about what type of physiological conditions that you need the activity like this, one of the most compelling, actually, changes that occurs on the ER is when cells encounter events that challenges ER to expand ER massively. And one of the examples was the B cell that I showed you earlier in my talk. But the other one actually is rather relatively old observation, where hepatocytes, the liver cells, their ER is treated by phenobarbital. The smooth ER in that case gets expanded. And so the long time ago, I think patients who are treated with the phenobarbital, turns out that their liver ER was pretty expanded. So we thought we would test if that actually has anything to do with this activity of ATF6. So to do that, we used ATF6 MEF, knockout MEFs, and put back wild type ATF6 and treated them with the phenobarbital. And I highlighted the ER with the red marks, and you can see that the ER is distended by the present treatment with the phenobarbital. However, when we actually put back mutant, this is the form of mutation that cannot respond to the DHS and DHC, treat with phenobarbital, the ER actually was not robustly distended. We are very excited about this, and obviously we created a lot of interesting questions, and being very new to this field, I really welcome all your suggestions and comments. But with that, somehow I can't actually hear. Just quickly summarize what I said today. ER stress induces those sphingolipids, DHS and DHC. And ATF6, one of the UPI initiator, can be activated through its transmembrane domain. And in the transmembrane domain, there is a sphingolipid, DHS, DHC binding motif. So with that, I'd like to thank the people who did the work of all these. Arvin Tan and Francisco Pina are very talented postdocs, and collaboration, proteomics was, lipidomics, was performed by Dan Nomura at UC Berkeley. And with that, I thank you for your attention. And if you have any questions or suggestions, I very much welcome you.